much about geography, but she doesn't know biology.
Required practical number 1, part 2. So, second part to our microscopy required practical is preparing wet mount slides. So I have some onion here and I've taken out a slice. We want a very thin peel of the epidermal tissue. So the best way is to grab the onion, split it slightly, take a layer and peel the inner part, which you can see actually there is very thin. So that's one layer of cells all working together, so it's epidermal tissue. Take a layer, like so, get a microscope slide, place your epidermal tissue flat as possible, try not to crease it, take some distilled water and put 1 drop onto your specimen, or 2 drops maybe. Okay. Take a cover slip, a small glass cover slip, make sure you've only got the one because they are very thin, and then using tweezers try not to get any air bubbles into your specimen. So actually lower it very slowly at a 45-degree angle. Yeah, don't throw it on like I've just done and make sure there's no air bubbles in there. Okay, if there's any excess water, use a paper towel, just dab it on the top. Okay, now you can't really see those cells under a microscope because they're see-through. Let's pop some stain on, some iodine. On one side of the cover slip, put a drop or 2 of iodine solution. Now you've got to draw the stain underneath the cover slip, so take your paper towel and actually pop it on one side of the cover slip, and it will draw the stain underneath and go over the cells. So give it a second or 2 and then take off any excess stain. The risk here, guys, is that iodine is an irritant, so if you get it on your fingers or your hands, make sure you wash them straight away. That is a wet mount slide for onion cells done. Moving over, we now have the equipment for human cells, wet mount slides, some cheek cells. So what we need to do is take a slide again. This time put the distilled water on first in the middle, 2 drops. Take a fresh swab to collect your cheek cells, make sure it isn't open. Open into the cheek area and really rub as many cells off as you can, like so. Okay, you should have enough cheek cells on the end of your swab now, and stir into the water to release those cheek cells onto the slide. Okay, this must go into the Virkon, which is disinfectant, straight away for obvious reasons. Place your cover slip on top. Again, try and do it slowly, not like I did, so you don't get any air bubbles. There you go. And then we need to stain the cells, but this time we're using some methylene blue on one side. Oh, you can see already it's starting to be drawn under the cover slip. Using your paper towel again on one side, try and draw it under. Get in there. And any excess, straight on top. This will stain your clothes, so try not to get it on you. Just be aware the other risk is glassware. If you drop anything, you can get shards of glass, so just be careful. Wet mount slide for cheek cells done. Let's pop those over here. Now we have our microscope set up. Okay, we're going to have the stage at the top again, lowest objective, and we will observe the onion cell slides this time. Same as usual, set it up, use your coarse focusing knob, change to medium power in this case, which will be magnification of times 100 in total. And then I want you to be using that to draw your specimen, like so. So I have some example onion cells under the total magnification of 100, and I'm going to try and find out the actual length of a cell in micrometers. Okay, now to do that is an equation. So we have length of cell is the length of the sample, which we're going to use a perspex ruler in a second, divided by the number of cells in the sample. So how do we do that? We take a see-through ruler, and it goes up in small increments of 1 millimetre. Now remember guys, 1 millimetre is 1000 micrometers, so that very small millimetre there is 1000 micrometers. We want to get that on top of our specimen now. So we take our slide, we pop our ruler on, any millimetre will do, viewing that under your 100 magnification. I want you to look down and I want you to count how many cells fit between the 1 millimetre or the 1000 micrometers. So I'm going to look, and actually I've got 1, 2, 3, 4, 5 cells fit between my two lines on my ruler. Okay, so let's go back to our calculation. So like we said, there's our calculation. Let's pop the numbers in. We had 1000 micrometer sample, that was between our 2 points and our ruler, divided by 5 cells that was in there, gives us an actual length of each onion cell is 200 micrometers. So let's write that on. Okay, now what's missing is a scale bar. So just to do that, we take another calculation, and it will be length of the drawn cell. So I take my ruler, I come over to what I've drawn, and I get my ruler in millimeters this time, so let's pop it that way. And let's have a look at just one of the cells. Let's say 94 millimeters, which we know we have to times by 1000 to get to micrometers. So we've got our 94,000 micrometers, which is the length of a drawn cell, times by 50, because our scale bar is going to represent 50 micrometers, divide it by our actual length of a cell, which we calculated before, so that's 200 micrometers, gives us an answer. Pop it into millimeters again, because it'd be silly to draw micrometers on our sample. And then over we go, 23.5 millimeters. Let's pop that on. There we go, that's 10, 20, 3.5. There to there represents 50 micrometers. That is our scale bar and our drawing done.